Hello students, I am Dr. Rajashree Mutha, MBBS DPB, Diploma Pathology and Bacteriology. Currently I am working at SMBT Hospital as a lecturer in Microbiology Department. So today we are going to study about the nervous system. So first we will see what are the learning objectives. So what we are going to study in this. So first, basic structure of nervous system is neuron. So all the things related to neuron, that what is the structure, function, and what is the classification of neuron. Then parts of the nervous system. So the nervous system is divided into three parts, that is central, peripheral, and autonomous. So that we will study in detail. Then meninges, that is coverings of the brain and spinal cord. Then ventricles, so these are the cavities in the brain which make the brain light in volume. Then cerebrospinal fluid, the fluid present in brain and spinal cord. Then brain in detail, all the parts. Then spinal cord, cranial nerves, spinal nerves, and autonomous system. So nervous tissue is a specialized tissue which transmits the stimuli. So in characteristics, neuron is a structural and functional unit of the nervous system. The cytoplasm present in the nerve cells is called as neuroplasm, and the covering is called as neuralema. Now neurons have lost permanently the power of division because they do not have centriole, so they do not divide, and the function of the neuron is transmission of nerve impulses. So here you can see the structure of neuron. So main thing is the cell body, that is also called as cyton, and then the fibers which are attached to the cell body, they are called as dendrons. They are the afferent fibers, that means the nerve impulse is traveling towards the cell body. And then the long fiber single, that is called as axon, so it is a efferent fiber. So efferent means going away. And the cell body contains granules, they are called as Nissel's granules, which contain RNA protein. The function of cell body is organizes and keeps the cell functional. Dendrons, they receive signals from other cells, and the axon which transfers signal to the other cells and organs. If this axon is having fatty covering, it is called as myelin sheath. It increases the speed of the signal. So the fibers are called as myelinated if the myelin sheath is present, and if the myelin sheath is absent, they are called as non-myelinated. The interval between the myelin the sheath, where the myelin sheath is not there, that is called as node of Ranvier. So it allows diffusion of ions, and the cells present in that is called as Schwann cell, which produces the myelin sheath. So this is in short the structure of the neuron, and the nucleus present in cell body controls the entire neuron. So this is structure of neuron and function of neuron parts. So here also you can see cell body, then short fibers are dendrites, and then long fiber is axon, and the cytoplasm present in axon is exoplasm, and the membrane is called as exolema, and the myelin sheath, that is fatty covering, is there. So the myelinated fibers are present in central nervous system, and non-myelinated fibers are present in autonomous nervous system. Now we will see classification of the neurons. So according to the structure, they are divided into three types: unipolar, bipolar, and multipolar. So unipolar, it gives a single fiber, single process, which divides into dendron and axon. The bipolar neuron, it has two processes, one dendron and one axon. Multipolar neuron, it has several dendrons and one axon, and it is seen in motor neuron of the spinal cord. According to the functions, they are divided into sensory, motor, and mixed fibers. So sensory neuron is called as the neuron which carries impulses from sensory organ like skin, eye, ear, tongue. So these are all sensory organs. So from sensory organ to the central nervous system, that is brain or spinal cord. So that is called as sensory fiber. And the motor fiber which carries impulses from central nervous system to the effector organ where we have to take the action, that is muscles. So it is called as motor neuron. And the mixed fibers, they contain both the parts of the sensory and motor fibers. Third type is according to the myelin sheath, they are divided into myelinated nerve fibers or non-myelinated nerve fibers. So this is basic structure of the or parts of the nervous system. So nervous system is mainly divided into three parts, that is central nervous system, peripheral nervous system and autonomic nervous system. The central nervous system is mainly of two parts, that is brain and spinal cord. Peripheral nervous system, this is the main contains is nerves which arises from the brain and spinal cord, so cranial nerves and spinal nerves. And autonomous system is composed of sympathetic nervous system and parasympathetic nervous system. The cranial nerves are of three types: sensory nerves, so they are 1, 2 and 8 in number. Motor nerves, they are 3, 4, 6, 11 and 12. And mixed nerves 5, 7, 9 and 10. So total 12 pairs of nerves are present in cranial nerves, which arises from the brain. Now we will see coverings of the brain. They are also called as meninges. So brain and spinal cord are surrounded by three connective tissue membranes and they are called as meninges. So cranial meninges, they are covering brain, and spinal meninges, they are covering spinal cord. The outermost covering of the meninges is called as dura mater. It is tough membrane. The middle layer is called as arachnoid matter, and the innermost layer, which is vascular and it is attached to the brain, is called as pia mater. Then the space between dura mater and arachnoid matter is called as subdural space, which contains the blood mainly. And arachnoid matter and pia matter, which contains the space, is called as subarachnoid space. Sub means below, so subarachnoid space between arachnoid matter and pia matter. So it mainly contains the cerebrospinal fluid. And the pia mater and arachnoid matter also called as leptomeninges. So ventricles, they are cavities in the brain which make the brain lighter in weight. So there are total 4 ventricles: 2 lateral, 3rd ventricle and 4th ventricle. So 2 lateral ventricles are also called as paracele. So they are present in cerebral hemisphere. Then both come together and form foramen of Monro. So that is interventricular foramen. Then it continues with the 3rd ventricle, that is also called as diocele, which is present in cavity of the diencephalon. Then it continues with the aqueduct of Sylvius. Then it continues with the 4th ventricle, which is present in the cavity of the medulla oblongata. And then finally this 4th ventricle continues with the central canal of the spinal cord. So in this way cerebrospinal fluid flows from lateral to 1 and 2, then to the foramen of Monro, then to the 3rd ventricle, then 4th ventricle, and then to the central canal of the spinal cord. Now let's study cerebrospinal fluid. So it is a clear fluid that surrounds the brain and spinal cord. So this CSF is produced by specialized cells, that is ependymal cells, present in the choroid plexus of the ventricles of the brain. And about 125 ml of CSF at 1 minute is produced, and about 500 ml is generated every day. The function of cerebrospinal fluid is to protect brain and spinal cord from the injury. Also it provides nutrients in form of glucose to the brain and spinal cord, and the waste they are generated, they are removed through the cerebrospinal fluid. So these are main functions of the CSF. So location of CSF, it occupies the subarachnoid space, that is the space between arachnoid matter and pia mater, and the ventricular system around the brain and spinal cord. So it fills ventricles, cisterns and sulci. Sulci means these are the depressions, as well as the central canal of the spinal cord. There is also connection from subarachnoid space to the bony labyrinth of the inner ear via perilymphatic duct, where the perilymph is continuous with the cerebrospinal fluid. And the significance of this is when there is leakage from inner ear, it may produce CSF rhinorrhea, means the CSF is throwing through the nose. It is called as CSF rhinorrhea. So this is a clinical significance of the CSF. Now the composition, so it is mainly derived from the blood plasma, but proteins are less in this. So CSF proteins mainly 15 to 40 mg%, glucose is 40 to 80 mg%. Normally RBCs are not seen in CSF, and WBCs, they are 0 to 5 per cubic millimeter. Thank you. So collection of CSF is done by LP method, that is lumbar puncture method, and it is also called as spinal tap. So here lumbar puncture needle is there. So physician will first clean the part selected, that is the space between L3 and L4 lumbar vertebra. L3 means third lumbar and L4 means fourth lumbar vertebra, the space between two vertebrae. So with cleaning with spirit swab, betadine spirit swab, the needle is inserted into the intervertebral space, that is space between two vertebra, L3 and L4 preferably, and then it will go into the skin, then bone, and then the cavity, that is CSF filled space in the central canal of the spinal cord, and then with help of syringe the CSF is collected, and then it is collected in a bulb for the examination. Now what is importance of CSF collection when we need the CSF collection? Because CSF collection is not a routine procedure like we do for blood collection. So it is not easy method for the doctor and also for the patient. It is painful procedure. So the indications or where CSF is required in the meningitis. So it is the inflammation of the meninges, that is coverings of the brain. So the meningitis can be caused by virus, bacteria, tuberculosis bacteria or fungus. So to know the whether in meningitis is there or not there and which type, CSF collection is required. Then in brain hemorrhage like subarachnoid hemorrhage, in brain injury or brain tumor, CSF collection is required to know the exact pathology. Thank you. I hope this session you will find useful. Thank you.